Hi, I'm here at ASH presenting on the enhancer deregulation in multiple myeloma. I know it's a very complicated topic, so if you don't mind, I'll just use a few seconds to explain what enhancer are before we dig into the topic. So enhancer are really small regulatory elements. They are essential for gene transcription. And when I say essential, are really the one dictating the gene they are transcribed and really dictating the cell identity in a specific cell. They are so important that normally, although they can regulate any gene in our genome, they are physically and functionally constrained to act in specific chromosome loop called insulated neighborhood. Because again, we don't want an answer to activate too many genes in our cells. However, in cancer cells, especially due to chromosome abnormalities, and these apply of course to multiple myeloma, this enhancer find a way to cross the boundaries and then activate genes. They are usually outside this chromatin loop and make them to become cancer genes. So focusing now in myeloma, we know that translocation, I'm thinking about the 414 translocation, can cause actually an enhancer to migrate from one area of the genome to another area, driving genes such as MMZ or FGFR3. Specifically myeloma, there is a group of patients, these are the non-hyperdiploid, they are known to have this IGH enhancer that actually get translocated to different genes. These are cyclin D1, but also MAF, and more importantly MYC, that we know how important it is in myeloma biology. Now, we know myeloma is characterized by this translocation. We know MYC is important, and we know that we can actually argue that this dependency of super enhancer of myeloma cells could also be what we can call an Achilles heel that we can actually use specifically for cancer cells to treat them and eventually to kill them. And we have right now some therapeutics, some way to target this super enhancer dependency of myeloma cells, and I'm thinking about BET inhibitors, these bromodomain inhibitors such as JQ1, or I'm thinking of about CDK inhibitors, for example. They are also in clinical trial. And these are all strategies that again can take advantage of the fact that cancer cells are really addicted to super enhancers. So if we can target that, we have actually a very selective way to kill cancer cells versus normal cells. And I will also finish mentioning that super enhancer seems to be important also for mechanism of resistance. We have our own data in the lab where super enhancer can cause also resistance to IMiDs and BET inhibitors. And I will argue that if we have really strategy to target these cells, this super enhancer, we'll probably have a way to restore sensitivity when resistance occurs.